For decades, clinical trial oversight relied entirely on isolated, manual documentation scattered across physical desks. Trial compliance was dictated by physical artifacts. Everything from the signatures of the principal investigator, the lead researcher at a site, to paper case report forms and static regulatory binder indexes existed in a vacuum. The shift to digital tools failed to solve the underlying architectural problem. Moving online merely replicated the analog mindset, resulting in dozens of disconnected spreadsheets. This creates severe operational friction. Clinical research associates, the professionals who monitor these trial sites, are forced to manually cross-reference patient screening logs against the trial's electronic data capture system, trapping data in dangerous silos. The solution requires a relational bridge. By linking the data underlying these files, a single update automatically cascades across all clinical, safety, and operational logs simultaneously. A true oversight dashboard shifts the operational burden from the human memory of the research associate directly into the system's architecture. This prevents the human errors that cause GCP, good clinical practice, compliance failures. We are going to engineer a 9-module, audit-ready oversight dashboard from first principles. This interconnected system operates on 3 specific tiers: master directories, subject workflows, and compliance tracking. This map shows the complete blueprint of our relational architecture. 9 distinct nodes connect to form a unified ecosystem. This engineering automates alignment with international regulatory guidelines directly within the database architecture. The core principle of the network relies on strict routing. Every single piece of data must have a defined submission path to ensure ultimate traceability. By abandoning scattered notes and flat files, clinical operations professionals build an environment where regulatory compliance becomes a mathematical byproduct of the workflow itself. Phase 1 of the build establishes the administrative base. The Studies module acts as the master directory for tracking unique protocol IDs and current study phases. These protocol IDs are then linked directly to the master site tracker. This creates a real-time monitor for operational statuses, allowing teams to see exactly which sites are in startup and which are active. This data table shows the master site tracker in action. Looking at the row for Site 1, we see an active status, Dr. Stephen listed as investigator, an enrollment goal of 10, and exactly 2 current enrolled patients. From here, we introduce the enrolled patients database, the core relational node, linking those patients directly to specific subject IDs. This provides complete subject lifecycle tracking, seamlessly managing the transition of patients as they move from screened to randomized or to withdrawn. The module uses smart data validation. If a randomization or screening date is missing, the system immediately triggers an automated quality control warning. Linking individual subjects directly to site-level goals provides an unassailable, real-time view of the recruitment funnel. No manual reconciliation is required. Phase 2 builds the clinical core. The schedule of activities, or SOA log, governs the timing of all trial compliance. Tracking specific visit windows is a massive logistical headache, particularly in oncology trials where a patient's schedule drastically changes between induction and maintenance phases. This timeline graphic shows how we automate that math. When a planned day is inputted, the system expands a bracket that generates the precise start and end date based on the protocol's built-in formula. Every specific required procedure, like an ECG or a tumor assessment, is mapped directly to its corresponding visit name. This mapping acts as a digital checklist for source data verification, the process where monitors check original medical records against the trial database to ensure accuracy. Automating these window calculations removes the primary vector for human error. A protocol deviation is never triggered by a simple math mistake. Phase 3 introduces risk and regulatory automation. We deploy the regulatory checklist as the digital trial master file, the primary tracker for all essential study documents. The system uses intelligent status tracking to conduct a real-time gap analysis of what is filed, pending, or collected in a site's regulatory binder. Automatic alerts flag time-sensitive documents, prioritizing follow-ups on expiring investigator CVs or training certificates before the next monitoring visit. Next, we attach the Safety and Regulatory Logs module, designed for the rigorous tracking of adverse events and serious adverse events. This safety log dashboard shows the system's automated critical deadline tracker. When an onset date is entered, it calculates the timeline, flashing a bright red overdue alert if missed. Event severity is also mapped automatically, isolating severe or life-threatening events. In the high-stakes field of pharmacovigilance, an automated deadline tracker transforms systemic reporting risks into highly visible, manageable tasks. Phase 4 completes the network with the closed-loop monitoring hub. The ultimate administrative engine is the monitoring visit log. This categorizes the monitor's workload by visit type, site initiation, interim monitoring, or closeout, and precisely tracks the mode of the visit, whether on-site or remote. Shows how we manage report submission deadlines using comparative logic. For Site S002's interim monitoring visit, comparing the planned date of March 17th to the actual date of March 22 instantly triggers an overdue flag for the final report. The protocol deviation log enforces dual date integrity. Finally, the action items database acts as the resolution engine. Opening that overdue visit record reveals a nested list of findings, including a critical CAPA task for recurring missing signatures. The database utilizes priority tags to force the resolution of critical issues, ensuring that missing consent signatures are addressed before new subjects can be enrolled. Every single task demands definitive ownership, explicitly assigning the responsibility to the monitor, the investigator, or the clinical research coordinator who manages the site daily. Embedding action items and protocol deviations directly into the visit log creates a self-contained engine where no deficiency can ever be orphaned. We can run a real-world traceability test on this network. During source data verification, a monitor discovers a systemic error. The site continually leaves the primary signature line blank. The automated escalation begins immediately. The monitor logs this missing signature directly into the site initiation report. Logging the finding automatically generates a protocol deviation. A reportability toggle is checked, teeing the issue up for immediate escalation to the Institutional Review Board. This nodal diagram shows the final step. The deviation originates at the visit log, passes to the protocol deviation node, and hard links directly into the CAPA module, locking the action item into a critical state. This exact, unbroken relational path is what a QA auditor or regulatory inspector inspects. True traceability provides mathematical proof that the system identified a clinical mistake and forced the implementation of a corrective plan. The traditional tools of trial management, the isolated notebooks and static documents, rely entirely on the limits of human vigilance. We replace that vulnerability with the rigorous, automated structure of a 9-module relational dashboard. File expiration alerts, automated schedule windows, and safety reporting deadlines ensure that no timeline is left to chance. Scaling a modern clinical trial requires moving beyond digital spreadsheets and embracing genuine systems architecture. To protect the integrity of clinical data and the safety of enrolled patients, your operational oversight system must be as mathematically sound as the scientific protocol it protects.